Okay, in meiosis, it is different than mitosis. It does have the same names for all the stages. There's still prophase, metaphase, anaphase, and telophase. But meiosis has two stages to it. So you still have chromosomes being duplicated, but in the end product, and either males or females, you end up with 4 cells. Even in females, it's a little different how you get the 4th cell in females, but the result of mitosis, you have 2 daughter cells. In meiosis, you end up, 1 cell ends up making 4 23 chromosomed cells. And I'll get to that. Yeah, there is duplication, but then meiosis 1 occurs a little differently than mitosis. Meiosis 2 is the exact same as mitosis is. So these two things, your big take from meiosis will be these two things. The number of chromosomes is reduced to half. You have to have that. You can't have a 92 chromosomed zygote. That's not a human being. 46 chromosome sperm cannot penetrate a 46 chromosomed egg. So there's a point when sex cells are forming that the number of chromosomes has to be cut in half. Now, there's another thing that happens. It's called crossover, and that's for genetic variability. And I have a really good diagram to show you for that. So let's go through all the stages. I do have some other videos for you to watch. I hope they help. I apologize for the sound. Okay, meiosis 1. So here we go. It says, during prophase 1 of meiosis 1, chromosomes coil up just like in mitosis. They condense. The chromosomes stick together in pairs. Now that is different. Gosh, I don't want to bounce around, but let me show you. I'll come back to this. See this representation of, well, and even up here, a little bit bigger picture. See how in prophase, how they're all scattered out, and it kind of looks like, well, they're sitting next to each other, or like here. They're really not. It's very random. And then metaphase, remember, they line up exactly on top of themselves during mitosis. We're doing prophase 1 of meiosis. They lined up as what are called homologous pairs. All the chromosomes pair up like they're in a karyotype. And remember, where is this coming from? You get 23 from the mom and 23 from the dad. I had mentioned 1 earlier. I meant for every single chromosome for traits, you get a single one from each parent as far as the 23. You get 23 chromosomes from the mom and 23 from the dad. So where were we? Okay, so we're on to prophase 1. Now, this is the part, it's called crossing over, and we're going to be very general with this. It's easier to understand that way. So within each set, chromatids, remember they're getting duplicated. The homologous chromosomes exchange crossing over segments. They cross over. It rearranges the genetic information. See how they're paired up like this? And what ends up happening is one part of a chromosome actually attaches to the other part of its homologous brother or sister, or they're homologous pairs. They're pairing up and they start sticking to each other. And that just seems so random for something that is so important. But when they do, little pieces break off and they exchange genes that way. Now I'm going to go to, I gave you this diagram. This is the best diagram I think I've found. I gave you a few of these. But let me come back up here. I got some long, skinny, and we'll go through that. This is spermatogenesis and everything. Great diagram. There we go. So let me make this a little smaller. And I think that's the only ones it is. Okay, so see these? Here we go. We have sister chromatids. Remember, this side, well, and they're kind of crossing these over. So this side, would represent 46. That side represents 46, just like in mitosis. Now, during meiosis 1, or prophase 1 of meiosis, see how these start sticking to each other like this? It says crossing over between non-sister chromatids, like that. These are the pairs, but it's saying non-sister because these are joined together by a centromere. So you have a centromere, and then you have these sister chromatids, and then you have another centromere here with these sister chromatids. And now you have the non-sisters crossing over like that. See how now there's a piece of red on the blue and a piece of blue on the red. And then when they split up, like I said, you get 4 cells. Now look, you have these two, and then you have these two that are different. That's variability. And it says recombinant daughter chromosome. Recombinant's when you exchange DNA or add DNA to a certain structure. Here's the best example that I have. And I'm going to leave it on this, and then I'm going to go back to our outline. So this is spermatogenesis. This is how, just a summary of meiosis. Here we go. Parent cell, maternal chromatids. Like I said, 23 for the mom, 23 for the dad. See this? It says gene for blood type, gene for eye color, gene for hair color. And it's just kind of showing all these genes are carried on these chromosomes. During meiosis 1, which what happens is eventually this cell is going to split into 2 cells. Do you see how the genes have been there's recombinant DNA? They've been exchanged. Here was, and it doesn't really designate it. Let's say this is the dad, the red. See, you're going to have a lot of dad's genetics, but you're going to have some of mom's as well in this particular cell. Same over here. If this is the mom, this is the dad. Here you have mostly mom traits maybe and some of the dad traits. It's a genetic lottery. So that happens during prophase 1. It's called crossing over. You have exchange of genetic information. Metaphase 1, and this is also different, they line up side by side in the middle of the cell. And this is prophase 1. These aren't exactly lining up. This is metaphase 1. See how they're next to each other? I mean, they still stack on top, but these homologous pairs, they stay stuck together. Remember, mitosis, they all lined up top to bottom. They weren't paired up in homologous pairs like this. So the sister chromatids are still attached at the centromere, where they're anchored to their spindle. You know, here's the centrioles. Those are the spindle fibers that are attached. They're attaching to the centromere, the middle part. And then what happens during anaphase 1 of meiosis, and I'll have to use a diagram. These are going to get pulled to opposite sides, but they're still like an X. So the blue's going to go that way, the red's going to go this way. And I'll have to show you in a diagram. And then telophase 1 and cytokinesis. Let me go over here. These are better. And I'll show you what this and the sperm part mean here in a second. This is actually pretty good. So there's meiosis. I know I have another one. I just kind of threw a whole bunch in here to you. Okay, well, let's do this one. So this is mitosis. Look, so there's prophase, and look, see how they're all spread out, and then here they're connected. That's why it says no synapses. Oop. Oh my goodness. Sorry. Let's go back. Okay, I'll just leave it on this side. So here's mitosis. See how they're all spread out? Like the blue and the red are not attached. This is mitosis. See, during meiosis 1, or prophase 1, I should say, see how they're all connected? That crossing over is happening. They're exchanging genetic information. The chromatids are. Here's metaphase 1. See how these line up straight in the center, you know, on top of each other? See, the homologous pairs line up side by side. And yeah, you still have the same number. Like I said, the number gets duplicated. You never hear this. A cell's about to divide. If it has 46 chromosomes, very temporarily, it's going to have 92. Like right here, you have 92, and then when it starts to split, you have 46 here and 46 here. Same situation here. They get duplicated. The homologous pairs will pair up and stick together, and then they align like this during metaphase 1. Anaphase 1, you see the entire thing is being pulled over like this, which in this case, see, it's splitting them like this. So you have 46 and then 46. These are still considered to be, even though they're paired up, these are still considered to be 46 chromosomes. They're just paired up. And you'll see, in fact, you'll see these referred to as haploid even, because there are 23 pairs. That's where the math doesn't add up. Telophase 1, see these splitting apart, and then you can actually see there's a blue attached to a red. There's a blue with a red attached to it, so you have some genetic recombination have gone on during meiosis 1. And then here's telophase, and that's cytokinesis. Now, here's meiosis 1, and this is showing a very brief, because it's just like mitosis. This cell and this cell, see, this one's going to split into these two, and then this one is going to split into these two. And each one of those is haploid. They have 23 chromosomes each, and that's why they have the single ones. It says they are not genetically identical to the parent cell, whereas mitosis, they are genetically identical. All right, so anaphase 1, homologous pair breaks. The chromosomes now migrate towards sides of the cell. It says, unlike during mitosis, during anaphase 1 of meiosis, the sister chromatids migrate as a pair to opposite sides. They don't get split apart. Okay, they're still paired up, and they're homologous pairs. Telophase 1 and cytokinesis, they divide. And then, like I was saying, you get two cells from this, and then they undergo what's called meiosis 2. So chromosomes coil and condense. The nucleus, nucleolus disappear. Spindle fibers form, just like with mitosis. They align in the middle of the cell. Anaphase 2, they are pulled to opposite ends. And then telophase 2, the nucleus, nuclear envelope reappear. The chromosomes uncoil, and now you have 4 haploid cells. Well, and this one kind of does the same thing. So look, it's a side-by-side. Here's mitosis. You see they're not paired up, whereas they are here in prophase 1. Here's metaphase. Here's metaphase 1. See, the homologous pairs are side-by-side. They're not stacked on top. That's the only difference. This blue would be sitting right next to the red. Okay, the numbers are the same. These then get split like this, whereas these end up 2 cells, 46 chromosomes each. That's where mitosis would stop. So these have 46 chromosomes. You can't do that with sex cells. Like I said, you can't have a 46 chromosome sperm and a 46 chromosome egg. So these are split, and then right here it says meiosis 2. These will then split into 4 haploid cells, each with 23 chromosomes. To me, and we're doing this very superficial, the biggest differences are they pair up here. See, they're not paired up. And this is where the crossing over occurs, the genetic variability. And then here, you see they're side by side, it says stacked. And then these are pulled apart. Remember, these have 46 chromosomes. So do these. These also have 46 chromosomes. They're just still paired together. And then when these split, you get 4 23 chromosomed cells. So if you do the math there, that ends up being 92. So same as here, you'd have 46 and 46. But mitosis, you get 2 genetically identical daughter cells. Here you get 4 haploid cells, and they are not genetically identical to the original cell. I hope that makes sense. I think after you do your lab, it'll hopefully reinforce that even more. Let me show you this. This is how sperm form. This is called spermatogenesis. This occurs in the testes of males. Now, these primary spermatocytes, there is actually a cell called a spermatogonium, which is one above this. Those divide by mitosis. That way, there's always a constant supply of those. And then when the sperm cells get ready to form, you start out with this 2N, 46 chromosomes. So here's meiosis 1, see the first division. This is where the crossing over would occur. And then here is meiosis 2. And you end up with, these look like tadpoles, 4 sperm from this one primary spermatocyte. In males, that happens puberty to death. It starts at puberty and then occurs throughout life. That's a good picture. I'll show you one more, and I included this one because it had these spermatogoniums. So here is that one I was telling you about. See the spermatogonium? It says mitosis. These are constantly dividing, so they stay here. And then these are selected to go through meiosis. And so there's the first meiosis, here's the second meiosis, and then you have 4 sperm from that. In females, there's a twist. Here is a primary oocyte or oocyte. See, in guys, this was a primary spermatocyte. Same idea. Now, in females, meiosis 1 occurs prior to being born, and it stops in what is called suspended prophase. It goes through prophase 1 and then stops. And then at puberty, it finishes the first meiotic division, and that's where you get what's called a secondary oocyte. So this starts here, from here to here would start prior to being born. And then right at puberty is when you get this division. You get a polar body and you get the secondary oocyte. I don't mean to bounce back and forth, but see, here's the secondary oocyte, and then it shows there's the polar body. It's a very uneven division in females. So that's the meiosis 1. This secondary oocyte, remember, it starts going down this way, and if a sperm is there, and if it occurs, that's how fertilization occurs. So here's where it's different. The second meiotic division in females only occurs if a sperm penetrates the egg. That's why that's showing this. If a sperm penetrates the egg, then what you will get is a zygote. You'll get the second division, the meiosis 2. You'll get another one of these polar bodies, and then you end up with a 46 chromosomed zygote. You still have 4 cells. Look, it started out with 1. Here was 2, and then if the sperm gets inside, it's going to divide again, and you get a zygote and a second polar body. So you still get the 4 cells. I hope these videos help. I might mention this in some directions to you, but just remember, in females, meiosis 1 starts prior to being born, and then it stops in what is called suspended prophase. The only way that females undergo meiosis 2 is if a sperm fertilizes the egg. It's not like that in guys. Guys are fertile myrtles from puberty to death. They produce 4 sperm from one primary spermatocyte, barring injury or disease.
The ovaries produce secondary oocytes, the female sex cells. During fetal development, stem cells, called oogonia, go through mitosis. Some of these cells develop into 46 chromosome primary oocytes. At the onset of female puberty, tens of thousands of primary oocytes each form part of a primordial follicle in the ovaries. Between menarche and menopause, each primordial follicle develops into a mature follicle, and its primary oocyte completes meiosis 1. Some primary oocytes become secondary oocytes, 23 chromosome cells that begin meiosis 2, then pause. Each month, one secondary oocyte is released into the uterine tube. Meiosis 2 is completed when the oocyte is fertilized. The resulting ovum can develop into a zygote.
I don't think I have. In fact, I'd put another diagram. Those are really good. I like simple diagrams. Here's one for sperm that are forming. See, it's got 23 chromosomes. I thought I had one. I guess I don't. Thought I had one for females, but that's okay. I think I'll put one for females in there, just another one. But that's just taking you through the first and second meiotic. Just another example of meiosis. Okay, let me go back up here. Yep, and I went through all these. So we went through all these diagrams. Here's mitosis one more time. You get 2 cells. Here is meiosis. You end up with 4 cells. 4 haploid cells. Now, I guess the difference would be a zygote is not a haploid, but you basically end up with 4 cells. So it says meiosis 2, the N. So the N represents, in our case, 23. But okay. All right. I'm going to stop it there because we've run out of material. But that is kind of an overview of mitosis and meiosis.